This is Chapter 6 of General Virology, and we are going to be doing an overview of the immune system and immune responses to viruses. So, obviously, the immune system is incredibly complex. We're just going to scratch the surface. I'm going to have this divided into two parts. In the first part, I'm going to talk about what's called the innate immune system. And in the second half of the lecture, I'll talk about the adaptive immune system, and then we can talk a little bit about how viruses can evade the immune response. But as you'll see, it can get very, very complex, and it's obviously critically important in terms of medicine and our ability to interact with pathogens. Um, so we do have a whole class in the biology department that's devoted to immunology, and I encourage you, if you're thinking about a career in healthcare, uh, to consider taking that class. All right, so if we think about this virus versus host interaction, um, it is a true battle, right? You've got the virus trying to gain access to an advantage over the host. So it's an immediate battle, but it's also a long-term, constant evolutionary battle. So, viruses are constantly changing, hosts are constantly evolving new defenses, and so it's sort of an arms race, if you will, right, where a host cell may evolve a new defense against a particular virus. The virus then evolves a way around that particular defense, and so forth. And so if you think of it from a virus's perspective, um, if a virus triggers a very strong immune response, it may get completely cleared from the host before it's able to be transmitted to new hosts. That would be worst case scenario for a virus, right? It's basically being defeated by the host's immune system. Um, but on the flip side of that coin, if you have a virus that is extremely virulent, so virulent that it quickly kills the host, that's actually not in the virus's best interest either, even though it's winning that immediate battle. If the host dies very quickly, then it's not capable of spreading the virus to new hosts, right? And that's the one thing every virus needs is a fresh host to infect. So really, and from an evolutionary sense, the most successful viruses are the ones that can escape just long enough to be able to replicate, effectively spread, right? Even if they're ultimately defeated, as long as they've spread to other people, that would be a success from the virus's point of view. All right, so in terms of the immune system, we're all at least vaguely aware of the immune system and the idea of immunity. Um, but if we think of it biologically, what is the immune system? Well, it's a set of cells, right? White blood cells, proteins, receptors that are also proteins, whose whole job is to sense and control infection. So our human immune system has these two arms. It has the innate immune system and the adaptive immune system. And I'm going to focus here in this part on the innate immune system, which is very immediate, is activated very quickly, as we'll see, but it's not particularly specific. And it includes cells such as macrophages, dendritic cells, natural killer cells, whereas the adaptive immune system, it takes some time to get going, but it is very specific for individual pathogens, and it's also very long lasting. So you can get immunological memory, production of antibodies that are going to potentially allow you to recognize that pathogen for weeks or months or years, depending on the particular instance. And the adaptive immune system is primarily revolves around T cells and B cells. So this graphic illustrates the relationship between the innate and the adaptive immune systems. So here on the x-axis, you're looking at the number of days after an infection occurs, right? Here's infection at day 0. And then on the y-axis, we've got the magnitude of that immune response. So you can see the innate immune system kicks in very quickly, right away. Peaks maybe about 4 days after infection. But the magnitude of the response is relatively low compared to the adaptive system, which is begins to be activated maybe around day 3 of the infection, but isn't really peaking until day 10 or 11. Okay, but it's a much stronger, higher magnitude response. And again, here we can see that some of the different cell types that are involved with those two different responses, and we'll uh, bump into these as we go along. Also wanted to point out here, aside from some of these cell types, we have chemicals called cytokines that are involved in both, but are primarily associated with that innate response. All right, so in order to kick on that innate response, somehow cells in the host need to be able to recognize that a pathogen is even present. And so this really comes down to receptor ligand type recognition. So host cells will carry what are called by virologists pattern recognition receptors, or PRRs. So these are receptors on the surface of host cells that will recognize viruses in this case. And the generic term for what they're looking to recognize are called PAMPs, which stands for pathogen-associated molecular patterns. So those, what does that mean? Well, basically means viral nucleic acid, RNA, DNA, or viral proteins, right, that could be recognized by these receptors on the cells. And there has to be some physical binding between those two. And the idea is once that PAMP binds to the pattern recognition receptor, that's going to activate the receptor, trigger a signal transduction cascade that is then going to eventually lead to pretty quick activation of that innate immune response. So just to talk a little bit about what these receptors are and what kind of things they recognize, there's really two different classes of receptors that recognize viral PAMPs. So there's what are called the toll-like receptors, TLRs, and the RIG1-like receptors, RLRs. So there's 10 different TLRs. 6 out of the 10 have been shown to recognize viruses. You have TLR2 and 4 on the cell surface, and TLR3, 7, 8, and 9 that are all found in endosomes. And that kind of makes sense, right? Viruses show up at the cell surface, and then as we saw in previous chapters, a lot of them get endocytosed and end up in the endosome. So these are both good places to recognize viruses early in an infection. The RIG1-like receptors are found in the cytoplasm, the red structures here, and are primarily recognizing nucleic acid, either single-stranded RNA or double-stranded RNA typically. So just to give you a little bit more detail, TLR2 and 4, which were the cell surface toll-like receptors, they usually recognize viral envelope proteins, right? So we're talking about enveloped membrane-covered viruses that have proteins extending from that envelope. And so these would be recognizing viruses that are in the process of attachment. Whereas the TLRs 3, 7, 8, and 9, as I said, are in the endosome, which is oftentimes maybe where uncoding might be happening. So they are more often than not going to recognize nucleic acid, double-stranded RNA, single-stranded RNA, even viral DNA sequences. So different specific TLRs are sort of specialized for different types of nucleic acid. And we can see very detailed, like crystal structures of some of those TLRs, but you can see here TLR3, for example, binding very tightly to some double-stranded RNA. And I wanted to include this table just because it's a nice summary of those different classes of toll-like receptors, what type of ligands they bind, and then you get some examples of different viruses that are known to be recognized by those specific receptors. Now, in the RIG1-like receptors, we said these are found in the cytoplasm, bind to RNA, either genomic or transcribed viral RNA, And there's three different types of RLRs. There's actually RIG1, for which the whole class is named, which recognizes virally transcribed single-stranded RNAs, but also short double-stranded RNAs um, from either negative sense or positive sense RNA viruses, and even from some double-stranded DNA viruses as well. Then you have MDA5, which recognizes longer double-stranded RNAs, primarily from positive sense RNA viruses, and LGB2, which seems to play more of a regulatory role. But just to point out before we go on, what you'll see here, for example, is these either TLRs or RLRs. Once they get activated, you'll see you have arrows pointing towards the nucleus, and you see inflammatory and type 1 IFN genes. Right, so this is really referring to cytokines. Okay, so this interaction between the host cell receptors, TLRs, RLRs, and those viral PAMPs is going to induce the expression of these small proteins called cytokines. So here you've got virus being recognized by the TLR in this case, right? That activates a signal transduction cascade, which kicks on the production of these small protein cytokines. These then get excreted and are sent out to be recognized by receptors on the surface of other cells. Those cells become activated. And it's basically, you've got cell 1 here recognizing the virus directly, and then it is sending off the alarm, right? It's sending out the signal, hey, guys, pay attention. I just found this virus. You need to be on alert. That's basically what's happening here. So common cytokines include like the interferons. You might have heard type 1 interferons, INF alpha, INF beta. These are two big antiviral cytokines. First discovered in the 1950s, and these were so named interferons because they were shown to be able to interfere with viral replication. So these interferons, these cytokines, will bind to the interferon alpha or beta receptor that is present on the surface of all cells, right? So basically any cell in your body is capable of recognizing these cytokines. And for that matter, pretty much any cell type in your body can also produce those type 1 interferons. So this is a pretty universal system. And the cells that are then responding to these interferons, they are activating genes that are specifically interfering with viral replication and primarily really with viral translation. So I know this looks like there's a lot going on here, but just to simplify it, so here you have your type 1 interferon binding to the interferon receptor, right? So this would be one of these cells, for example, right? Imagine this is the interferon receptor. Here's the cell producing the interferon, so it's getting that signal. Go back. So that activation now is going to trigger basically the production of these two proteins, PKR and OAS. And so you can see what PKR is doing on the left side of the cell, and OAS on the right side. But essentially, PKR is going to end up inactivating this EIF2 alpha, and that is going to have the effect of blocking translation. So essentially just shutting translation down. So not only is viral translation going to be blocked, but also cellular proteins won't be made, at least temporarily. The other protein, OAS, is activating this RNase enzyme, and RNases are enzymes that digest RNA. So it basically chews up single-stranded RNAs. So this would include cellular messenger RNAs, also viral messenger RNAs, and if you happen to be infected with a single-stranded RNA virus, right, it would chew up the genome of that particular virus. So both of these, even though it is going to affect cellular activity overall, it's going to slow down viruses' ability to complete the replication cycle. So quick, rapid, first line of defense, but very non-specific. And while we're talking about cytokines, I wanted to bring up this idea of cytokine storms because this has gotten a lot of press lately with COVID. So the very worst cases of COVID-19 seem to involve activation of what are called cytokine storms, which can be life-threatening in and of themselves. And so the whole idea here is you're getting these waves of cytokine activation, right? Primary cytokines activating cells that are then producing secondary cytokines. Um, and this can essentially be like a positive feedback loop where you're just getting too many immune cells activated, too many cytokines being produced. They're very potent signaling molecules. So you're getting large amounts of cytokines, and this can lead to systemic inflammatory response syndrome, um, among a few others, acute respiratory distress syndrome. You essentially end up in a situation where your immune system is now attacking your body and not just infected cells. Um, and so that can lead to organ failure and death. So pretty serious. All right, so hopefully you're getting the picture of the innate immune response being quick, rapid, virus binds to receptor, triggers production of these cytokines, which are going to have this hopefully uh, repressive effect on viruses' ability to complete the replication cycle. And so just to tie in with some of the cell types that are associated with the innate response, so we've got macrophages, which are large immune cells. They're phagocytic cells, meaning they can essentially eat other particles or cells in the body. Macrophages are found in all parts of your body, in all tissues. One common job is just to remove red blood cells that are uh, not in good shape or that need to be replaced. But they do also have receptors on their surface that can bind to antibodies that would be coded on invading pathogens, including viruses. And so this, you're going to see macrophages tied in with the more specific adaptive response in that respect, and we'll come to that. You also have dendritic cells. So these are what are called antigen presenting cells, or APCs. And they're also phagocytic, so they can eat antigens, which is what you see happening in this image here. So this is a dendritic cell. You can see some antigens in green being ingested by that dendritic cell. And they can then actually present the antigen or part of the antigen on their cell surface and essentially show it to other cell types in order to activate them, which again we'll talk about here in a second. I just was talking about them being antigen presenting cells. Hopefully you're all aware of what antigens are, but just to remind you, right, these are small molecules that are capable of causing an immune response. And then another cell type that you see associated with the innate response are what are called natural killer cells, which induce apoptosis in infected cells. But to really get into what they're doing, so they sort of tie in with the adaptive response as well. So we'll kind of circle back around to those. But just keep in mind they are technically activated in the innate response. But we will revisit here in a few minutes. So here's kind of the cartoon version of how dendritic cells would work and begin to bridge the gap between the innate response and the adaptive response. So here's your cell, right? You've got your virus comes into the cell, is establishing an infection. You've got viral antigen, whatever that might be, viral nucleic acid or more likely viral protein. And dendritic cell sees this, ingests it, and it then enters the lymphatic system, travels through lymph vessels to a nearby lymph node, and it will then, as I said, present that antigen. So you can see it's formed this special structure on its surface where it's actually holding out that antigen to, in this case, a T cell. So it physically presents it to the T cell, says, "Hey, I found something weird, out in this tissue where I was hanging out. Take a look at it. What do you think?" And that antigen presentation can then activate those T cells. Once they become activated, they will actually leave the lymph node and enter the bloodstream, return to the site of infection, um, and as we'll see, that can then start triggering an adaptive immune response to that particular antigen in those infected tissues. So bottom line, um, we get this quick non-specific response, production of cytokines, which trigger production of proteins in nearby cells that can hopefully repress viral replication. We have dendritic cells ingesting viral antigens and using those to activate T cells, and that will lead us into this discussion of the adaptive immune system in part 2.